Okay, so for chapter one, we turn our attention to a rather interesting group of organisms. These are the fungi. Now, may not really think much about them. You walk by them in the woods, perhaps something that looks like this. This has a neat name, a name to it. This is actually known as, common name is turkey tail. These, you see them a lot growing on trees. They almost look like brackets, or sometimes called bracket fungus. But that's turkey tail because it kind of looks like that. The one up above, Lactarius, is really pretty. It's a blue color, which is, I think, kind of the interesting thing about fungi. We can get all kinds of different colors and variations out of them. They're a very odd group of organisms, but we're still sticking with the idea of spores as being a part of reproduction. That's what that little part of the drawing is right there. Lactarius indigo does truly have that blue color to it, and it's got kind of a sap, a white sap that's kind of sticky. So it's sort of an interesting fungus. Let's talk a little bit about fungi in terms of comparison to some of the other critters out there. There's probably about 1.5 million fungal species, and they come in a variety of types. There are single-celled versions. Yeast, for example, is a fungus. That's a single-celled version. There are multicellular versions, like the one you see in the background. This is the honey mushroom, and these grow all over Florida as well. I see these all the time. They're very common. These are a little bit lighter in color, but the honey mushrooms that we see down here in Florida have much more of a honey color to them, hence the name. But interestingly enough, when people always want to know what's the largest single organism on the planet, is it a blue whale, or is it a redwood or anything like that? People overlook the fact that the largest single organism on the planet is a singular honey mushroom complex, just an organism that covers somewhere in the vicinity of about 9 million square meters of forest, and is somewhere in the vicinity of 1,900 years old, and it weighs hundreds of tons. That's the world's largest organism. It's a mushroom. Now, if you're looking at that honey mushroom in the background, and you're thinking, oh my God, what it would be like to walk out in the woods and see a mushroom that's that size, right? You think you would be able to see it from space. You might learn as we go through our discussions today of fungus that most fungal growth is underground. So this organism that we're talking about that is this size, 9 million square meters, most of the organism itself, most of its body structure is subterranean so that you don't see it. That's how they know it's the same organism. They take a sample from a subterranean area of the mushroom over here, and then they move some distance away, and they take another sample from the mushroom or from the fungi in the other place, and they compare them genetically to find out if they're exactly the same organism or not. And sure enough, hold on one second. And sure enough, it's one critter. So anybody ever ask you what's the world's biggest critter, it actually is a fungus. They reproduce sexually, they reproduce asexually. That's most typical in many species because they use spores. And remember, we talked about the fact that spores in other species are very good for waiting for the environment to change. So if you don't have a very conducive growth environment right now, a spore can be a resistant structure waiting for the environment to change to a point, and then that spore can germinate. So remember, we've been harping on the idea that organisms have to adapt to their environment, but sometimes adapting to your environment might mean something like a little bit of patience, just waiting around until the environment becomes better for germination, for growth. Now, the way we get our food, or the way organisms get their food, is very different than fungus. For example, the way we get our food is we basically bring it into our bodies and we digest it inside of our bodies, in our stomachs, obviously. These organisms do a little bit differently. They engage in something we call extracellular digestion. And what we mean by that is fungi extract nutrients from their surroundings by secreting these exozymes. The exo part of the exozymes refers to enzymes that are secreted outside of the body. So unlike us, where we bring the food into our bodies and then the enzymes in the stomach mix with the food and digest it, in a fungus what they do is they secrete these exozymes out into the environment, and they digest the nutrients in the environment. So think about these fungi living in this sort of like a soup of digested nutrients, and then they just simply absorb those nutrients from the environment. So very different. It makes them very good at being decomposers too, which we'll look at here a little bit later on. Our closest ancestor is the fungus. Animals and a fungal ancestor lived somewhere between about 460 to 670 million years ago. When I was showing you the different protozoan diversity I mentioned to you at the time, if you look on that, we are more closely related to a fungus than we are to plants or any other eukaryote out there. And fungi make a pretty good size, a pretty good chunk of the diversity of organisms on the planet. As we might expect, the insects rule the planet in terms of diversity, quite a few different species of insects. But the fungus have a pretty good size of the pie, too. So this is not a small group. Very interesting group of critters that's able to survive in all these kinds of conditions. Nutrition-wise, like I said, they secrete exozymes. They use something called a mycelial mat, which I'll show you in just a minute, but that's how they digest their food. And mycelial mats, as you'll see in the pictures, have very high surface area. And if you remember from your Bio 1 discussions as to why cells remain small, cells stay small because they have a high surface area to volume ratio, and that makes them more efficient. More efficient in the sense that things can move across the membranes more efficiently. So if you think about what I've said about the fungi, it secretes these exozymes into the environment, nutrients are digested, and then the nutrients are absorbed by the fungus. Having that very high surface area means an awful lot of nutrients are being absorbed. As I mentioned also, these are really good decomposers. They are some of the only critters that are known to be able to break down cellulose. Now, there are some prokaryotes that do that too. Cellulose, remember, is the indigestible carbohydrate that makes up the cell walls of plants. We can't digest cellulose. Cows eat cellulose and actually can use it as a form of energy, but that's only because they have a symbiotic bacteria that lives in their gut that actually helps them break down cellulose. Cellulose is not something that's very easy to break down. And even more so than that, fungi are also good decomposers of a substance known as lignin. This is the chemical formula for lignin over here in the lower left. Again, lignin is a really complex, very strong, very durable type of molecule. And it also, if you look at all those rings of carbon, it's a very rich molecule in terms of carbon. Carbon is a foundational element. You learned in Bio 1 for all living organisms out there. So fungi act as really good decomposers and recyclers because they can break down these huge carbon sources like cellulose and lignin and a variety of other organic materials and liberate that carbon back into the environment so other organisms can actually take up that carbon and survive. Us animals, thank goodness for these decomposers, we can neither digest cellulose nor lignin. By ourselves, we can't digest cellulose or lignin. As I mentioned, the cows do, but they do it with symbiotic bacteria. And I'll show you here in just a couple slides, there are even some carnivorous fungi out there, which is kind of interesting to think about. All right, what is this mycelial? Well, in order to understand what a mycelial is, and this is very important in terms of how these organisms grow and reproduce and get their nutrients, we got to take a look at first the body structure. The body structure of fungi are made of these things called hyphae, and hyphae are long filaments, and they might be one long filament that's one cell, or it could be long filaments of several cells. If you look down below, I'll show you the two types. There are the septate hyphae, and they look more cell-like, right? Each one has a nucleus. And then the cenocytic hyphae on the right-hand side. There's no cell walls at all. So in the fungi that have cenocytic hyphae, it's one giant cell and a bunch of nuclei. Even in the septate hyphae, even in the ones that have the septate hyphae, you know, the septate hyphae, notice they have pores through them. So materials can move through those cell walls or through those septate very quick. And what this allows for, this either cenocytic or the holes in the septate, is it allows for very rapid growth. Fungus are very much opportunistic. So as I said with the honey mushroom, most of the structure of that critter is underground. And when it's time to reproduce, that's when they pop up above ground. The toadstools that you see from the honey mushroom, the caps, if you will, that's just a small part of the organism. So that's why you're walking out in the woods, you're not going to see a 9 million square meter toadstool. It's the stuff that's all underground. It's these uh, filaments of hyphae that are running through the ground, and they're secreting exozymes, and they're bringing in food. Well, being synocytic or being septate, the septate having the pores, means that when it's time for growth, that growth can very rapidly occur, because what fungi will do is cytoplasm will quickly stream or flow towards that end of the hyphae. So if I could get you to kind of imagine a bunch of cytoplasm streaming into this part of the hyphae right here. My pen always. So we get a bunch of cytoplasm streaming into the hyphae right here. And if you imagine that's kind of like a balloon blowing up a balloon. So as that cytoplasm rushes into that area, eventually that balloon or that part of the hyphae would expand and eventually could form something like the toadstool, right? The actual mushroom itself, and that would push itself above ground. I've heard it said before that when it comes down to fungi, the real entity in a fungus is not the cell, it's mostly the nucleus. Fungi are more concerned about the nucleus than they are about the cell being in a nucleus. So that's why we have things like these synocytic hyphae, which basically is one giant cell with a bunch of nuclei. Multinucleate, having more than one nucleus in a cell, is a very common thing that happens among fungi. All right, so we got these little long, slender filaments of hyphae. And if we put a whole bunch of those together, there they are all underground. There's a bunch of hyphae right there. And they're extending down. So this is the thing that would be 9 million square meters in the honey mushroom underground. So like I said, they went over here and they took a genetic sample, and they went to the other side and they took a genetic sample, and they found out there was actually the same individual. The reproductive parts, or the toadstools in this case, those are only going to come about when conditions are right. And as I said in the previous discussion with the hyphae, the way they do that is when conditions are right, right? When the environment is right for producing spores, for reproducing, basically, cytoplasm is going to stream through these hyphae, and it's going to plump up those hyphae and form that mushroom. If you've ever looked at a mushroom and torn apart with your fingers, you'll notice that it's basically layers, and those are layers of hyphae laid down on top of one another. So that would be a bunch of those little tubes, synocytic or septate, that have all been mashed together. A group of these hyphae, the part that extends underground, is called a mycelium, or a mycelial mat. And if you take a look here at this picture, this is all, wood debris and stuff, but all of the white that's growing through here, these are all hyphae. These are all part of the mycelial mat. Lots and lots and lots of surface area, right? Lots of surface area means lots of absorption of the nutrients in the environment. Here's some more pictures of hyphae. Here's some hyphae in the upper left. Now think about this, the surface area to volume ratio stuff here. This is 10 microns. So look how many hyphae you can find in a small area like that. And then in the background, this is some hyphae I got a picture of. This is hyphae. See how it spreads through the environment? Here's some pictures of synocytic and septate hyphae down below. So mycelium is all these hyphae connected together, growing through the substrate, increasing their surface area and allowing them to absorb. Now, interestingly enough, I said this a couple of slides ago, we're more closely related to fungi than we are to plants. And one of the things, one of the properties, and one of the characteristics that unites us with these organisms is the cell walls of fungus are not made of cellulose like they are in plants, but they're made of chitin. Now, I don't know if you remember chitin from Bio 1, but chitin was a polysaccharide. It's a specific polysaccharide. One of the places that we find it predominantly is in the exoskeleton of insects, like this male rhinoceros beetle that I've got a picture up here. So that's kind of interesting, right? So that's one of the connections that kind of unites us more closely with the fungus than it does with the plants, would be the fact that they have chitin in their cell walls, and some of us, insects most prominently, also have chitin. I told you there were carnivorous ones as well. Carnivorous fungi, what they basically do is use their hyphae as kind of like a a lasso, and so this nematode, which is an animal, a worm, a roundworm, very small, not very big. We're looking at 25 microns here. These hyphae can actually swoop around this worm and actually do it pretty quick. The loop can close in something in less than a second. And so you think about this hyphae ties this roundworm up here, and then more hyphae grow over here, and basically it entangles the worm in all those hyphae, and then the hyphae start secreting those exozymes, and there you go. That's it for the worm. So you don't have to worry about walking your dog through the park and having a big toadstool attack your dog. It's not that kind of carnivory, but carnivory of small, soil-like structures like we find in nematodes. Excuse me a second. Goodness, the weather turns cold, and already I feel like I'm catching something. Fungal hyphae can also parasitize plant cells through what we call haustoria, which is plural, or haustorium. The fungal hyphae is the yellow up on top, and then the plant cell wall. The fungal hyphae just penetrates through the cell wall, and then the hyphae moving, or the haustorium, which is the hyphae moving into the cytoplasm of the cell to be able to digest the nutrients. Probably not, only because the hyphae aren't that strong. You would think something a frog or a mouse would. Let me tell you, if there was ever a fungus that was big enough to do something like that. Now, if you talk about something after it's dead, that's a little different story. But to catch something like that, that'd be a pretty scary fungus. I mean, we're talking about carnivory down here at the very microscopic level, right? You see these little tiny roundworms. That's a good question, though. Neat to think about. Okay, well, now there are other carnivorous fungi. Fungi make their living with the exozymes. This also gives them a good opportunity to be good symbionts, right? Or symbiotic relationship, remember, is two organisms living in a relationship with one another. Either one gains a benefit and the other one is hurt, or they both gain a benefit. I've got a little review of this if you've forgotten. But the type of fungi I want to talk about now are called mycorrhizae. And mycorrhizae are a fungus. This is the furry stuff you see here. These are roots of plants. Those are the roots. Mycorrhizae form a mutualistic relationship around the roots of plants. It's so important, this mycorrhizae, that on 90% of all vascular plants, you'll find this fungus growing on the roots. What it does is it extends the surface area for the absorption and the digestion of nutrients. Remember, the fungus is secreting exozymes to digest nutrients for itself. But out there in the environment, in that subterranean world, it's secreting a bunch of these exozymes, and so lots of stuff is being digested. So the roots of the plants that the mycorrhizae are growing around, they gain the benefit of this nutrient soup, if you will, right? The pre-digested organic material that the fungal hyphae present for the roots. So as I said, very important. There's a really interesting visualization of that. Up above are two species of the same plant. The one on the left was sprayed with a fungicide, which would kill the mycorrhizae, which is a fungus, and the one on the right was not. You notice the difference in growth between those two plants. The one on the right does significantly better if it has those mycorrhizae. Like I said down below, it's an extension of the root system. More surface area, more soil contact, more absorption. Now here on campus, we have a very interesting plant, and this is a plant. It's called Indian pipe. It has little tiny flowers. Indian pipe is white. It does not photosynthesize. So not all plants, not all flowering plants, not all plants photosynthesize. Indian pipe, which I went and looked the other day here on campus, and I couldn't find where they usually grow, but maybe they're usually earlier in the fall. Indian pipes are involved in what's called a tripartite relationship. Indian pipe parasitize the mycorrhizae that live on the roots of the tree. So it's a three-way relationship. The mycorrhizae are living around the roots. They provide nutrition for the roots of the plant. The plant provides structure for the mycorrhizae to grow on. The plants also provide some carbon for the mycorrhizae to grow, and then the Indian pipe comes along and actually grows on by parasitizing the fungus, the mycorrhizae fungus. So that's kind of neat, right? A white-colored plant. You can imagine some of the first investigators looking at that. That looks like a flowering plant, except it's missing one very big thing that we know about plants is how does it survive? You know, how does it photosynthesize? Mycorrhizae come in two types. There are ectomycorrhizae, which grow on the outside of the root. So down here, this is the cross-section through the root. Hopefully you recognize these as the tracheids, the xylem and the phloem. And all the blue on the outside, that's the fungus. That's the mycorrhizae. See, it grows all around the roots and then in between the cells. The other type is our puscular mycorrhizae. Our puscular mycorrhizae doesn't grow around the root, but grows in between the cells. It penetrates into the cell wall, as you can see right here. This is one of those Haustorium or pusculars, not Haustorium. But it doesn't penetrate the cell membrane, so it doesn't affect the cell negatively. Right, so this is a good example of what we would call a mutualistic symbiotic relationship, right? The fungi gain a place to live, and they gain a little bit of carbon, and then, of course, the plant, like I said, gains a much more efficient way of extracting nutrients from the environment. All right, now I said there's a lot of weird things about fungi, but one of the probably strangest things has to do with the way they reproduce. So let's take a look at sort of a general way of reproduction. Asexual reproduction does occur in fungi, and again, that's rather typical. And asexual reproduction is a very simple. You have a spore-producing structure. That would be the toadstool, the actual mushroom. If you think about that structure, they produce spores asexually. So they produce identical clone spores. The spores germinate, grow into a mycelial mat, back to spore-producing structures, all asexual, okay? If conditions warrant sexual reproduction, that's where it becomes a little bit different. Two mycelial mats, so the hyphae from two mycelia will attract one another. They do this through a thing called pheromones. Pheromones are externally released chemicals like hormones. They relay a signal. So two mycelial mats will use these pheromones, these chemical signals, to attract one another to prevent inbreeding. So in other words, the pheromones, a mycelial mat would avoid a hyphae of the mycelial of itself with the same pheromones, but it would be attracted to the hyphae in a mycelial mat that's secreting a different kind of pheromone. So that's how they recognize themselves from other organisms, if you will. But here's where it gets a little bit weird. When those mycelial mats come together, the first thing that happens is the cells fuse at the cytoplasm level, not the nuclei. So we get something called plasmogamy. Plasmogamy is just the cell fusing. Remember I said that in fungi, it's about the nucleus, not so much about the cell. So what we have here, the part that's in the purple there, is an area that we would call a heterokaryotic stage. Heterokaryotic stage means that in a cell, if the fungi has cells, the septate one, the nuclei are not fused. You have basically, in some organisms, you might even have two haploid nuclei in a cell. We would call those organisms dikaryotic. Now, they're not diploid. Remember, diploid is the nuclei fuse. But in these dikaryotic or these heterokaryotic, which would be more than two, you have cells, or those long filaments of the syncytic hyphae, that are all separate nuclei. If they do have cells, like in the ones that are septate, you might have two haploid nuclei in a cell. Now, what is the advantage of that? Well, the little asterisk shows you. Being heterokaryotic, you have some of the advantages of being diploid, right? You got two haploid nuclei per cell, so you have some of the advantages of being diploid. That means you have another set of genetic information. Now, does that mean that being diploid is better than being dikaryotic? Not necessarily, remember? Just because an organism does it a little bit differently, just like when we were talking about the haploid structure of moss gametophytes, doesn't mean that that's not successful. Fungi have been around for 500 million years, and they've been working with this heterokaryotic stage, And so it's not a matter of diploid or heterokaryotic. It's a matter of diploid works here, heterokaryotic works here, works just fine for these organisms. Now, plasmogamy and the next thing that happens, which is karyogamy. Now, karyogamy is when the nuclei fuse. Those might be separated. So that heterokaryotic stage can sometimes last for a period of time. It can last hours, in some cases days, months, or even centuries. Being the diploid, the advantage of being diploid means that during this heterokaryotic stage, this organism has a better chance, has a better set of genetic information to deal with the environment with. All right, now if the environment is conducive, then karyogamy occurs. The nuclei fuse and we form a zygote. Now from our point of view in the animals, the zygote then undergoes mitosis repeatedly to make a multicellular adult. Right? When you developed, the sperm and the egg came together, and that was a fusion of the nuclei. It was a single cell, and then that grew into multicellular, and of course, Junior was born. But the zygote in most fungi is very short-lived. It's basically the only diploid stage, and it's basically made only for one reason and one reason only, and that's what you see in the box that follows the zygote part. Right after the zygote forms, it undergoes meiosis. Now, I write in a question mark there, purpose. What's the purpose of meiosis again? What kinds of cells does meiosis produce? And okay, but that's right, but what types of cells in terms of genetic information? Are they clones of one another, these cells, or are they different? Think back to your meiosis in Bio 1. What did you end up after meiosis 1 and meiosis 2? You made sperm and egg, but were they all identical to one another like you would think in mitosis? Or were they different? No, they were all different, weren't they? They're all genetically different. Remember I told you earlier, the purpose of meiosis is variability. The purpose of mitosis is quantity. All right, so we're going to produce these spores through meiosis. Now, these are little different spores than the ones that were produced by asexual reproduction. I guess what I should say, you can produce spores sexually or asexually. So when this zygote undergoes meiosis and produces these haploid spores, those are produced through sexual. And then they germinate and they form a mycelium, and either they engage in asexual reproduction or they engage in sexual reproduction. So if we compare animals and fungus, asexual reproduction is a lot less common in animals, a lot more common in fungi. Animals, as far as the ones I know, don't have a heterokaryotic stage. They don't have a plasmogamy followed by some time in the future by karyogamy. I mean, there is plasmogamy, that's when the sperm and the egg come together, but pretty quickly the nuclei fuse. Here we're saying that plasmogamy and karyogamy can be hours or days even apart. And then, of course, like I said with the animals, once the zygote forms in the animals, that's the multicellular thing. That's going to become multicellular, it's going to make the adult. Here it lasts for just a short period of time. Its only purpose, folks, was to put those nuclei together, have them run through meiosis, prophase 1, crossing over, all of the variation that you get so you can produce a bunch of different types of spores. Questions on that? Well, we'll look at the reproductive cycle in each of the major groups of fungi when we get into them, and we'll go ahead and revisit this idea of being heterokaryotic. Many species are sexual and asexual. There's about 20,000 species that belong in a group of fungi that we call the deuteromycetes. Now, this isn't a term that I've seen used everywhere, but it is a term that is still occasionally used. We classify fungi, as you'll see as we go through the different groups, based on the type of reproductive structure they make during sexual reproduction. So when we want to know what group of fungus this is, and we'll get to the groups here in just a moment, we first need to find out what type of sexually reproducing structure it makes, okay? And that tells us what group it fits into. The deuteromycetes We've only observed asexual reproduction in them. Now, if they sexually reproduce, we haven't noticed it yet. So we throw them into the Deuteromycete group, which is, years ago, I remember when I was in college, they used to call the Deuteromycetes the imperfect fungi. Not because there's something wrong with them, but because we don't know exactly what group they fit into because we haven't observed the sexual stage. Molds fit into this group of asexually reproducing organisms, like you see the lovely gray stuff that's growing on the orange there. That's Penicillium. Under the microscope, Penicillium makes these little things that look like little fingers, or like the Hydra, you know, the Greek mythology with the snakes coming out of their head. And all of these are spores that are being produced. Yeast is another example of an organism that can asexually reproduce. This is Saccharomyces cerevisiae, the same yeast we use to make beer. And you can see we've got a parent cell and then budding. And there's some budding going on there. So they divide just basically divide through asexual reproduction. Remember the thing about asexual reproduction, though, folks, it produces clones. Those are clones of one another. All right, a little bit of the ancestry. The oldest fossils from fungus are somewhere around 460 million years ago. I don't really have to tell you too much about the difficulty at finding fossilized fungus. But when we look at the molecular clock analysis, we think that animals and fungi diverged, or fungi arose on the scene somewhere between about 1 billion to 1.5 billion years ago. So we're missing about a half to a million to a million, or 500 million to 1 billion years of evolution of these organisms for reasons that I've mentioned. Right? They just don't fossilize very well. Soft-bodied critters. We do know that the ancestor was aquatic, unicellular, and flagellated. Maybe something like this organism you see over on the right. These are called the nuclarids. That's a nuclarid. The fungi and the animals are very closely related to one another. So here's some aquatic, unicellular, flagellated ancestor. There's the animals and the rest of those. Remember how the protozoan group is polyphyletic, so they're all mixed in. Nuclarid, for example, is a protozoan. All right. The fungi and the animals are closely related to one another. They form a group that we know as the opisthokonts. Kont is a pole or flagellum. Opistho means it's behind. So like you see in the little picture there, these are sperm cells, right? Animals, we make flagellated cells that are opisthokonts. They have the flagella in the back. If you think about Euglena, Euglena has its flagella in the front. So nuclarids also have their flagella in the back as well. So again, when we look at organisms that have similarity of development, I've mentioned this a couple times this semester, similarity of development oftentimes indicates ancestry, some level of ancestry. Most fungi are actually more closely related to the nuclarids than they are to other unicellular, to animals, of course. The protozoans are in all these groups everywhere. All right, so we're going to look at the chytrids, very small group. The zygomycetes, bread mold is that group. A very small group, but very important, of glomeromycetes. And then the big groups are what we call the sac fungus, the ascomycetes, and then the club fungus, the basidiomycetes. About 30,000 species. This is the sac fungus. Looks like a sac. And basidiomycetes get their name from the reproductive structure. Looks like a club, but very often basidiomycetes are the ones with the toadstool, which kind of looks like a club. So let's do the chytrids first. There's only about 1,000 species of them. They're ubiquitous. What does it mean when something is ubiquitous, by the way? We might say, for example, humans are ubiquitous. I told you that chytrids, members of this Chytridiomycota group, are found everywhere, even some species down around those hydrothermal vents. There you go. Yep, ubiquitous means these organisms are found pretty much everywhere. Like I said, even around some of those harsh conditions like the hydrothermal vents. Chytrids act as decomposers. Anything that's secreting those exozymes is a great decomposer, right? Because when you secrete those exozymes in the environment, you digest materials in the environment. So you're decomposing it for other things to take up. They can be mutualists. They can be parasites. This chytrid here is parasitizing this Odygonium, which is a green algae. It's parasitizing that Odygonium. Chytrids very early on diverged from the fungal evolution. They have flagellated spores, which we call zoospores. That root zoo is often one we associate with animals. Now, these aren't animals, but we use the root zoo to oftentimes refer to something that's mobile, that moves. So these spores, some spores don't have a flagella, but the ones that do are able to move, so we call them zoospores. And there's one down below. And this is unique to this group, these flagellated spores. Notice once again the flagella on the back, the pistil cones. All right, the black bread molds. Black bread molds not only on bread, it's the gray fuzzy stuff that grows on strawberries. That's the same thing. There it is on bread. Let's start down here with the asexual. That's the most typical. Black bread mold, what happens is a spore germinates, mycelial grow, and then those mycelial grow a stalk, and at the end is a sporangium. Now, terms very similar to what we're looking at in the plants, right? With the sporangia, the megasporangium and the microsporangium. So these sporangium produce spores, and they produce lots of spores. And not only do they produce lots of spores, they disperse those spores. This right here in the picture, this is a fungus known as Pilobus or Pilobus. And they can actually turn their sporangium and face them in a particular direction. Ubiquitous is a relative term, Sean. We might say that human beings are ubiquitous across the terrestrial aspects of Earth. Or we could say that a species of fish is ubiquitous in the ocean. It's kind of like the word endemic. You can be endemic to Florida. You can be endemic to the United States. So it's a context. Like we couldn't say humans are ubiquitous in the universe or in the solar system either. See what I mean? That's for sure. Rhizopus forms these little lollipop-shaped things, these little sporangium. There's what it looks like when it grows on bread. And like I said, what comes out of there is tons of spores. And those spores will then germinate. And we don't have sexes in fungi. We have mating types. So two different mating types. The hyphae will grow, and eventually they come together. And they form initially something called a gametangia. And a gametangia, you can see right there in the middle, a gametangia is a fusion of hyphae with these haploid nuclei. So here we go back in this idea of plasmogamy without immediate karyogamy. So when those gametangia with those haploid nuclei fuse, you get the picture that you see down below there. Right there, right? So the cytoplasm part of the cell has fused, but we're heterokaryotic. We're N plus N, which means we have two. We're not 2N because we haven't fused the nuclei. We're heterokaryotic, so we're N plus N. Now, what happens is that gametangia is going to grow into a zygosporangium, a heterokaryotic zygosporangium, and that zygosporangium is going to grow, and it's going to become well, I guess the best way to describe it, it becomes kind of gnarly looking. It becomes very tough looking, like you see in the picture. It's got these bumps on the outside. It's a very durable structure. It's resistant to freezing. It's resistant to drying. It's metabolically inactive. Metabolically inactive. In other words, waiting for the environment to become conducive. This in zygomycetes, in these black bread molds, this karyogamy, these formations of these zygosporangium, are going to occur when the environment begins to deteriorate. So when the environment becomes bad for these organisms, they're going to form these metabolically active zygosporangium, very resistant, all right, that's going to stay that way until the environment becomes conducive again. And when the environment then becomes more conducive, now there is the idea the environment never becomes conducive, right? Some of these die. They form these zygosporangium, and the world never gets any better for them. So obviously they can't survive forever. They might be able to survive for weeks or centuries, but they can't survive forever. There's just no way, right? So some of these organisms are going to die off, and then other ones are going to undergo karyogamy. And then karyogamy is, again, a short-lived multinucleate zygote. All right, so the little purple balls in there represent the nuclei, and they're the fusion of each pair of those red and green ones from above. So it's a multinucleate, short-lived zygote, which undergoes meiosis for variability, which then produces a sporangium, which then produces spores. So the spores that are produced by this route, I don't know if it's a button I'm hitting or what, are going to be different from one another because they were produced sexually, right? We get karyogamy and crossing over. The spores that are produced by this route are going to be the same. They're going to be clones. So as I said to you earlier, we identify or we classify fungi based on their reproductive structure. We call these the zygomycetes, because they make a zygosporangium. It's like a spore-producing structure that's like a zygote. Let me get rid of all my scribblings, right? When karyogamy occurs, which basically produces a zygote. Oh, let me go back one more. So this is a group of about 1,000 molds, parasites, commensals, organisms that live without causing damage or benefit to the other organisms. Syncytic hyphae, so they can grow very quickly. There's no septate between them. All right, the other group are the glomeromycetes, very small group. These were once put in with the zygomycetes. Nearly all of these form that type of mycorrhizae that we talked about, known as the arpuscular type. Matter of fact, there's a picture. This is a plant cell wall here. There's the cell wall. And this is the arpuscle. This is hyphae is what that is, into the cell wall there. Not into the cell membrane, but just into the cell wall. So nearly all of the glomeromycetes form these arpuscular mycorrhizae, which are found on, like I said, this type. There's the other type of the ectomycorrhizae. Over 80% of all plant species have arpuscular mycorrhizae. Here's the root again. Right, the thing, and this is all mycorrhizae. Very important, like I said, without the mycorrhizae. And you can think about plants growing and moving out on the land. Remember, we talked about how harsh that would have been for the early plants. Well, one of the things that benefited them was moving out on the land with these mycorrhizae, because the mycorrhizae gave them a little bit of an advantage to help them much better digesting the nutrients in the environment. Could have. Yeah. Yep. As a matter of fact, that's what it would have done, because that's why we would have found that they were the same on either end of it. When we did the genetic analysis, they have to be clones of one another. So that has to be the result of asexual reproduction. Yep. Same critter. The ascomycetes are the sac fungi. Very often the name, the sac fungi is obvious. That looks like a sac down that red one on the lower left. These are morels up here. Morels are sac fungi. Each of these little pits is a sac. Most of the species of fungi, about 65,000 species, belong in here. These are bread yeast. molds, truffles, and things called morels. There are morels. This is a truffle. Truffles are very expensive. It's amazing. I've never had one, but I guess they're supposed to taste so good. Here, for 8 ounces, it costs you about, what is that, $600, $573 for 8 ounces of flash frozen black winter truffle. You know, they find truffles, right? They use pigs to do it. Truffles give up the same scent as male. They give up the male pheromones from a pig. So a female pig looking for a male, secreting its pheromones of sexual receptivity, will dig around in the root litter until it finds the truffles. And of course, the guy behind, he didn't want the pig to eat the truffles, so he gathers them up. Lichens, many times the fungal part of a lichen. Remember, I've told you a couple of times before that lichens are a symbiotic relationship between a fungus and some sort of a photosynthetic organism like a green algae or maybe a cyanobacteria, could be prokaryote or eukaryote. But often about a quarter of the species of the fungus that makes up the lichen are sac fungi, ascomycetes. Now, the term ascus comes again from the reproductive structure they produce. So let's go asexual again real quick. Asexually reproducing spores are called conidia in this group. Right? So we have sexually reproducing spores, which are just spores, but if they're conidia, if they're asexually reproducing spores. And conidia are formed on conidiophores, which are extensions of the hyphae, the mycelium. And each one of these is going to form a spore. 2 different spores of 2 different mating types are going to fuse, right? And they form something called an ascus. So there's plasmogamy again, right? So we get the fusion of the cytoplasm, and they form this thing called an ascus. There's the ascus right there. In this case, the ascus is dikaryotic, which it can remain for periods of time. This allows for more genetic recombination. And what we mean by that is as these little ascus are forming, different nuclei are ending up in different asci, right? So the same 2 nuclei aren't going to end up in an identical asci. There'll be many asci with different combinations of nuclei. Then karyogamy occurs and we form a diploid nucleus. So now we got a bunch of different types of zygotes. And then those short-lived stage again, the zygotes are going to undergo meiosis, right, which is variability. So, and you remember from meiosis, 1 cell makes 4, and then each of those cells are going to undergo mitosis. And so what we end up with, these little finger-like projections, these asci have 8 ascospores inside, like the little picture you see there. Now, the ascus is the reproductive part. The sac is the ascus. The ascocarp, the ascocarp is the reproductive part. Ascus means sac. And the asci are the little fingers that are on the inside. And inside each one of those little fingers are 8 ascospores. Those are going to be spores that were produced sexually. So they're different. They're different than the conidia. Conidia are asexual spores. Ascospores are sexually reproduced spores. All right? And again, whether they're going to engage in sexual reproduction or asexual reproduction depends a lot on the environment they live in. All right, the club fungi. These are the familiar, the mushrooms and the toadstools, puffballs. This is a puffball. These are spores. It looks like gas, but those are spores. And what they do is these spores are, they puff out of this fruiting body, and of course they disperse off into the environment. These are very important as wood decomposers. That's why you often find them growing on trees like that. Some species form mycorrhizae. Some are pathogens on plants. Like, for example, this is on a maple, I think. The black stuff, this is called a rust. Right? And this is called corn smut. It kind of looks nasty, but we'll look a little bit later on. Corn smut, this is actually a delicacy. You can eat this. It's supposed to taste pretty good. Some really cool club fungi, too. this is a mantia. This is the one from what was it, Alice in Wonderland? You know, where the Cheshire cat is sitting on top of. This one I think is really pretty. The maiden veil fungus. Those turkey tail fungus that I showed you earlier, those are club fungi too. All right, how do these folks reproduce? Well, they don't typically asexually reproduce. These kinds of organisms are more inclined to be sexual reproducers. So let's start in the left with two different mating types of mycelia. Because of pheromones that attract them, those mycelia then fuse together. Plasmogamy occurs, and when plasmogamy occurs, what we end up getting, this is a septate hyphae. Notice there's little walls between them. We end up getting a bunch of dikaryotic mycelium, right? See, these are all dikaryotic. They have two nuclei. And those dikaryotic mycelium are going to grow, and eventually it's going to outgrow the mycelium that only has one nucleus. So you see how these hyphae have both nuclei, where these only have one? So they're going to fuse, and then what predominantly is going to become the hyphae is going to be the dikaryotic ones. And those are going to remain that way for a period of time, allowing more genetic recombination is just a way of saying genetic mixing, right? Getting that variability. And then all this is going underground, right? At the right time, the cytoplasm is going to flood down through those hyphae, cause this, the end of the hyphae is like you're blowing up a balloon to plump up, and this toadstool or this club-like structure is going to come up out of the ground. And it can can occur very rapidly. This is a basidiocarp. It's an N plus N because it's heterokaryotic. Basidiocarps then form basidia on the gills. If you looked on the gills of a mushroom under a microscope, and if you look right up here, these are the gills of a mushroom. You know, the gills that stick out this way. So the blue part is the actual gill. This is the basidia. And basidia are N plus N. They undergo karyogamy. They make diploid nuclei. That's as far as the diploid part lasts. They immediately go into meiosis. The basidium then form 4 haploid nuclei. This is a basidium. This is where they get their name club fungus, because the basidium kind of looks like a club, right? And then the basidiospores. So the basidiospores are the sexually reproducing spores. All right? Lots of surface area on those gills. They can produce billions and billions of spores for reasons that I'm sure you all know about, right? More spores, better chance they survive. Now, I said these things can grow rather rapidly. In other words, these toadstools can pop up, almost overnight. You probably experienced this. You look out in your yard after maybe a rainy evening, and there's all these mushrooms everywhere. And it's like, well, where did they come from? Well, they were already there. They were all underground in a mycelial mat, and sometimes they actually form a ring called a fairy ring. The fairy ring name comes from ancient folks, with no other better way to explain it, thought that some fairy came down and sprinkled, I don't know, mushroom dust or whatever the heck they would have done while they were dancing around, and since they were dancing around in a circle, all that dust made these mushrooms end up forming in a circle. But you probably know better, right? You know that what happened is the spore germinated, say, somewhere right about here in the middle, and then the mycelial mat grew out concentrically, right? That mat of mycelia is growing. Now, if you think, why does it grow over here? Well, maybe local nutrients aren't very good for mushrooms to grow there. And when those mycelial mat grew out to that extension, and then when it was time to reproduce, the cytoplasm flowed to the ends of the mycelial mat, the hyphae, and then boop, popped up above ground. And since that mycelial mat had grown out concentrically like that, you get this pop-up of these toadstools all in one place. Sometimes you don't see a full fairy ring, too. Sometimes it'll be like, part of it, half of one. And that, again, that's going to depend on a lot of environmental conditions, but, the ground conditions. Fungi in the environment act as decomposers, as I mentioned. They break down cellulose, lignin. They recycle really essential nutrients like carbon and nitrogen. We talked about how important that is for plants. They can be mutualists, like the mycorrhizae. They can be mutualists with algae, cyanobacteria. They can be mutualists with plants or in animals. They can be parasites. They can be pathogens. So here's that symbiotic relationship thing again. I think I showed you this in an earlier slide. Remember that the name depends on what is the result to the species. So one species parasitizes another. Species A, in the upper left in that chart, gains a benefit. Species B, it's a negative. Commensalism is one gains a benefit, the other one is neither affected. Mutualism, they both gain a benefit. Competition is negative and negative because remember in competition, it means that nobody gets everything. So even if you get 99%, you still didn't get 100%. So still is a little bit of a minus there. Some of these fungi that engage in these relationships are obligate. In other words, they have to live that way. And some of them are what we call facultative. So they enter into these kinds of mutualistic relationships or symbiotic relationships. Not every time, just when it's necessary. Here's some mutualistic fungi. Mycorrhizae we already talked about. There's some mycorrhizae right here in this picture, sticking out from the plant cell, extending the roots in the surface area. There are the endophytes. These are fungi or bacteria that live inside the plants. These are usually sac fungi, ascomycetes. Very common, found in all plants. One of the things they can do is actually provide herbivore protection or pathogen protection by making toxins. You remember, plants can't run away from a predator. You know, some herbivore making its way through the environment, there's not much a plant can do, but a plant can make itself distasteful, right? Unpalatable. So what these fungi will do is they will live in the plant. The plant provides them a place to live, provides them with carbon, might even provide them with some food sources as well through photosynthesis. And the fungi turns around and protects the plant by producing these toxins that anything that come along that eat the plant would be affected by. Animals can make use of mutualistic fungus. For example, in the case of herbivores, there's many times fungi that live in the gut of herbivores that help them digest herbivorous material. These leafcutter ants engage in something called fungus farming, man. This is so cool. Leafcutter ants are known that because they take their big mandibles and they're able to cut out these leaves. So they take these leaves and they bring them back to the fungus, which they then feed to the fungus. The fungus, with its hyphae and exozymes, digests the leaves, and then at the tip of the hyphae, they produce these very nutrient-rich hyphal tips, and then the ants come and feed on the hyphal tips. So the ants are farming the fungus, and the fungus are turning around providing food for the ants. Like I said, sometimes this is an obligate relationship. It's kind of neat, isn't it? Think about organisms that enter into relationships like that. Like the pine cones, how? Yeah, same idea. That's right. Protect yourself by not becoming edible. Some animals would do that too. Monarch butterflies. Birds eat monarch butterflies. They taste bad. Okay, lichens. What is a lichen? Well, you notice here I got 15,000 species, but I put species in italics because we talked about species a few chapters ago. This sort of defies what a species is. A lichen is an obligate mutualistic relationship between a fungus, usually a sac fungus, and some, as I said, photosynthetic critter. So it's an obligate mutualistic. What does the fungus do? The fungus can protect the photosynthetic organism from the light, right? Many times the fungus have a color to them. And they'll protect the photosynthetic organism from the light. You might say, well, isn't light good? Well, maybe too much light, right? You want just the right amount of light. The fungus can also protect the photosynthetic organism from drying out. The photosynthetic organism, on the other hand, the cyanobacteria or the green algae, or in some cases it's both, provide actual food for the partner, right? Because they photosynthesize. Lichens are known as pioneer species too, because they can be found in really harsh habitats. We find them in the fossil record from as far back as 420 million years ago. The reason why this sort of pushes the idea of what a species is a lichen is two separate species that live together obligately. They have to live together. They've been mutualistic and been depending on each other for so long that if you tear them apart, they would die. You can see, like, for example, in these two pictures on the right and in the middle, and that great one down below, you can see the algal cells, the green photosynthetic algal cells, and you can see all the hyphae forming around there. They're also known as indicator species. Indicator species are species that are very sensitive to environmental disturbances. So, for example, if a botanist was concerned about a group of trees that might be suffering from air pollution from a nearby factory, many times it's the lichens that are going to start showing the damage first. So the lichens are, no, keep going with your comments, Phil. Sean, that's no problem at all. I like the comments. So the lichens will actually show the environmental disturbance first, right? Mean by an indicator species before the tree does. You might have heard a story years ago about a canary in a coal mine. Coal miners used to bring canaries down into the coal mine because canaries are very sensitive to carbon monoxide and methane, which builds up in coal mines and is, of course, can be fatal and deadly to the people working down there. But because of the way birds breathe, which we'll talk about when we get to the bird chapter, the canary is going to die first from these poisonous gases before the humans. So they bring the canary down in the coal mine. He's an indicator. When the canary dies, time for everybody to get out. The Cerridea here, that's how they asexually reproduce. So Cerridea is going to be a fungal cell with a little bit of, an algal cell with a little bit of fungal hyphae along with it. And they'll drift and find some other place to survive. But this is really kind of neat if you think about what we've described as species. These are actually two different critters. Not necessarily. I mean, they're good at, because they're kind of self-contained, because they've got a photosynthetic part, right, the algae, and they've got the fungus, which allows them to grab attachment, and then the hyphae can also attach nutrients. They're good at making do in environments when other organisms wouldn't usually survive. So don't confuse a harsh habitat with a habitat that's being changed or disturbed, right? So a lichen could be a pioneer species and grow in harsh habitats, but if that harsh habitat changes, right, these many times will be the first species to show. Does that make sense? Do they reproduce in conjunction with one another? Or do they mostly asexual reproduction, Philip? Mostly they engage in asexual reproduction. Now, there are all kinds of species of lichens out there, but we break them down into three groups based on appearance. There's fruticose, foliose, and crustose. Crustose lichens, and all of these grow on trees here in Florida, look like spots of, like crust. Sometimes people think of it as paint. Like here's a crustose lichen. Looks like spots of paint. You might have actually seen those sometimes when you're out walking around the woods. It looks like paint. I'll come back to this in a second, like this one right here. This is a Florida one. The Baton Rouge lichen. There's the red that we were talking about. That's the fungus. All right, that's a crustose lichen. The foliose lichen. Folio is like a portfolio, is like paper. It's leafy. So foliose lichens are leafy-like this one down here. That's a foliose. Another foliose would be this one here in Florida. This is a very common blue-green color lichen, a foliose, Anthoparmelia, that grows on the trees all over the place. That's a foliose. And then the other kind is fruticose. Fruticose is shrub-like. That's fruticose. This is fruticose. You walk up on a tree and kind of really look closely at it, you'll see all these different kinds of lichens. That's fruticose. What we call deer moss, which is actually a lichen that grows on the ground, that stuff there, that's actually an example of a fruticose lichen as well, of a shrub-like one. So those are the lichens. Fungus. Fungus can also be parasites and pathogens. About a third of all known fungal species are parasites or pathogens affecting mostly plants. Some attack food crops. About 10 to half of the world's fruit harvest is lost to fungi, like the tar spot on the maple leaves, or again, that corn smut. Corn smut, which is the scientific name is Eustallago matus. Corn smut is used to make a dish that's popular in Spanish cultures, and I got the pronunciation just because I had no idea how to say the word, but it's Huitlacoche, right? Huitlacoche. So that's actually a delicacy. It's supposed to taste pretty good. So it's basically corn that's got a fungus growing on it. Wheat fungus, right? Mushrooms. Claviceps purpurea. Claviceps purpurea is an ergot. An ergot is a type of fungus that grows on rye. The interesting thing about this is years ago when they used to have the Salem witch trials, and you remember maybe from school the way they did that, you would throw the witch in the water, and if she drowned, then she wasn't a witch, and if she swam, then she must be a witch because they threw her in the water maybe tied to a tree, or in worst case scenarios, they would burn these witches to the stake. And many times the reason why they were considered witches because they were acting strangely. They were having visions or hallucinations. Well, ergot growing on rye produces the substance LSD. So what people were doing back then is they were eating rye bread, and if it had been sat around for a while and had this fungus growing on it, they were actually getting dosed with LSD, and so that's why they were acting strange. And so these poor people were not devils or witches. They were basically just having a bad hallucinogenic trip. The chestnut blight, which wiped out a ton of chestnut trees here in Florida. I got this map. The chestnuts used to extend way from up in New England down almost into the southern part of the country. The chestnut blight, or the fungus, was brought into New York in the early part of the 1900s. By the time the 1940s came around, most 50 to 99% in this picture of the chestnut trees were dead. That's what a chestnut tree looks up above. Chestnuts roasting on an open fire, all that sort of stuff. We don't have a lot of chestnut trees. But you have just a few stands of chestnut trees, groups of chestnut trees anymore. So these fungal things can move very quickly, and they can be rather detrimental. You're thinking in 30 years wiping out most of the chestnut trees here in the United States. Trees are affected. Frogs are affected. This is Batrachiotritum dendrobatidis. This is something called chytridiomycosis, which is actually affecting frogs. That frog there on the right-hand side. You can see in the picture, these are chytrids, and this is a pathogenic fungus that's killing off frogs. This is causing, like I said, a worldwide decline of amphibian populations. They're either declined or extinct in about 200 different species. The big map that you see of the world, that's where this chytridiomycosis has been reported. This is a real concern among people who study these organisms because it spreads so quick. If you look down there in Mexico, you can see it was first reported up in the northern part in the early 70s, but by the time we get to the year 2000, about 30 years later, it's already spread down through most of the Central American area there, and still is problematic among the amphibian populations. And if people wonder why we would care about amphibian populations, you think about amphibians who feed on insects, and if you destroy one part of the environment, you obviously cause ill effects in the other part, right? We have to deal with certain fungi, human and animal diseases. Yes, it absolutely could. And in some cases, there's suggestions that some amphibian species have already gone extinct from this. Yeah. Things like fungal infections, and that moves so quickly. That's the problem. We suffer from yeast infections, candida, which is an opportunistic, causes something called thrush or vaginal infections. Many times a female, if she has is taking antibiotics, in the vaginal region, there are a lot of beneficial bacteria that live there and keep the yeast from growing, either by destroying the yeast or by directly competing for resources. And so if the woman's on antibiotics, she kills off a lot of the bacteria, and then the yeast can actually take effect. So that's a picture of candida on the right, and then down below in the green is a picture of what candida looks like under the microscope. Ringworm down on the lower right, that's ringworm, what it looks like on the skin. This is what it looks like if you took a sample. This is the skin. It's a sac fungus. Ringworm is not a worm. It is actually a fungus. Pneumocystis jirovecii, which is a fungal infection that can cause pneumonia in AIDS patients. It's a very rare form of pneumonia. You know, it's kind of one of those things where you hear about the person with AIDS who died of a rare form of pneumonia. You think, oh, this poor person, you know, they got AIDS and then they catch this rare form of pneumonia. Pneumocystis isn't so rare in the environment. It's just with a healthy immune system, it's pretty easy to fight off. But with AIDS, of course, you have a compromised immune system. So we also get affected by these guys with athlete's foot. Nail fungus, you see all the commercials on TV, and like I mentioned, ringworm too. They're not all bad though, right? Fungus are good. We eat them for food. We use them to make cheese, alcoholic beverages, bread, you know, yeast, of course, when they anaerobically respire. They make penicillin. If you take a look at that picture of Staphylococcus, right, that's a prokaryote, and that white part is penicillium. The thing that was noticed by folks who were working on these organisms is that area of inhibited growth. I don't know if you know the story about penicillium. That's right, because you're spreading those spores everywhere. Every time you scratch and touch, yeah. I had a friend of mine who did it. He was changing a tire in his car, and it was raining out, and he was laying down. Yeah, he was laying down on the wet grass and just picked it up there. He said it was a devil to get rid of. The guy who discovered the antibiotic properties of penicillin was growing some bacteria on a culture plate and left them on the window of his lab and went home for the weekend or the evening. And when he came back, some penicillin mold spores had flown in from the outside. His window was open. This was before air conditioning, it was a long time ago. And the penicillin grew on the plate. And he noticed that zone of inhibited growth, and that led him to do further investigation and found out that, oh, penicillin is a good antibiotic. As I mentioned, we use them to ferment carbohydrates like Saccharomyces. Very important in genetic research, too, like in genetic engineering. We can actually take fungal species and genetically engineer them to do things like produce insulin, which, again, is kind of like I was talking with the E. coli. They don't need the insulin, but they produce it, and we can then use that for people who need insulin for, you know, diabetics and stuff like that. That's penicillin up here. These are all little spores that radiate from the top. Really neat looking little, under a microscope, really cool looking organism. Alrighty, folks. Anybody got any questions for me? All right, well, I'm going to stop the lecture right there. I'm done for the day.